Having established how we activate our CD4 positive T cells, it's time now to look a little bit at how we are going to drive differentiation of our helper T cells down the various subsets. Keep in mind that these cells are essentially the center of the universe when it comes to the adaptive immune response, because they are essentially going to orchestrate all of the effector mechanisms that are going to be used to eliminate the pathogen. So think about it this way. If I activate a T helper cell, what are the other cells that they're going to interact with? Well, they're going to interact with B lymphocytes, which is going to drive their differentiation and isotype switching. So a T helper cell is going to essentially increase antibody production and secretion. Our T helper cells also interact with macrophage, and depending on what the response is, I can activate an M1 macrophage or I can activate an M2 macrophage. And also recall how dependent our CD8 positive T cells are on the T helper cells for the IL-2 that will help their proliferation. Differentiation to these various subsets is essentially driven by the type of infection an individual is having. So what's happened is, since they've started discovering more and more about the different T cell subsets, is that it actually makes a lot more sense. I remember when I was first learning about helper T cells, you were trying to fit a lot of information into this nice little package, and I got to tell you, the information tended to spill over and wasn't very precise. So if you actually had immunology more than like a year or so ago, what you're going to see is that a lot of this information has changed. So don't get uncomfortable, don't get upset, because actually this is way more straightforward than it has ever been. With all of these different subsets, there's a couple of things that I want you to keep in mind. They are all activated through the same process. You need your primary signal, and again, that's the T cell receptor binding to its specific pathogen via an MHC class 2. We are dealing with CD4 positive T cells here, and they all require the secondary co-stimulatory signal of the B7 on the APC binding to the on switch, the CD28 on the T cell. For the step 1, we really are going to focus on 3 primary subsets of T helper cells. And actually, these are going to comprise a majority of the helper cells that are going to be essential in the clearance of our different pathogens. So what we're going to be talking about today is differentiation down the Th1 pathway, the Th2 pathway, and new to some of you is going to be the Th17 pathway. And always go back to the same thought, that the type of pathogen is going to determine which one of these pathways are taken. Once you know what pathogen is involved in the type of infection, you will essentially know that you are going to either have a predominant Th1 response, a Th2 response, or a Th17. And we all know that we like to think in very concrete terms, very black and white. Well, if it's this, it only can be a Th1 or a Th2 or a Th17, which I've got to tell you, that way of thinking is going to be great for the step 1. I want you to keep these very delineated in your minds. So once I look at the type of pathogen, I pretty much know what pathway, and we'll talk about that on the next slide. The type of pathogen is also going to determine what cytokines are produced in response to it. And those cytokines with the helper T cells will essentially stimulate the production of unique transcription factors that are only going to be found in these subsets of T helper cells. This is one of those figures that you look at and either you say, I'm going to dive right into this and I'm going to memorize it, or most of us go, you know what, that's too much information right now. I'll come back to it at a later date. So what we've decided to do is actually break this figure down into its separate parts. If you want to use the figure as a whole, it's a great little review figure. But from this, what I want you to do is recognize a couple of things. First of all, any CD4 positive T cell prior to activation has the potential to become any one of these three subsets. Upon activation, though, it has to make a decision. And remember, that decision is based upon the pathogen, the body's cytokine response to those pathogens, and ultimately the transcription factor that is going to be initiated or upregulated due to those specific cytokines. So again, you sort of have a theme going on there. Another really important thing that I want to point out here, that a lot of different review books say different things about this, but I want to be really clear when I say that regardless of what subset you are dealing with, they all produce IL-2. And hey, Dr. Moscatello, do you remember what IL-2 does?
I don't know, but ask me when I'm 85 and following you in a walker.
Okay, she's a little bit older than I am. She's forgetful. Anyway, so remember that IL-2 causes the proliferation of our T cells. So I'm not going to talk about IL-2 in these figures. I assume you recognize IL-2 is produced by any of these subsets. So let's break it down. Let's go ahead and start at the beginning. It's a very nice place to start. We are going to first talk about the pathogens that drive cells to differentiate along the Th1. And this is fairly straightforward. Actually, out of all of the three pathways, Th1 is probably the most straightforward. So when I have an intracellular infection, and we're talking about viral infections here, or any of our intracellular bacterial infections, the body responds by making a fairly significant amount of IL-12. And it's back again. Remember, IL-12 is going to be responsible for the activation of our NK cells, but here it's serving a secondary role. It is going to drive differentiation of my CD4 positive T cells down the Th1 pathway. And the IL-12 is essentially going to turn on a unique transcription factor that is known as T-bet. Okay? And the T-bet is then going to activate the unique genes associated with a Th1 response. And I hope you recall that one of the things that Th1 does really, really well is that it will produce and secrete very, very high levels of interferon gamma. So what we need to do now is focus on what is the effect of this interferon gamma that has been produced. So very quickly, if I have an intracellular infection, I'm going to make lots of IL-12. That IL-12 will induce the transcription factor T-bet, and T-bet in turn is going to activate a Th1 response with lots of interferon gamma production. So what does that interferon gamma do? Well, it actually has several roles. One of the things that it does, though, is simply amplifies a Th1 response. So it is essentially going to backflow and positively influence any CD4 positive T cell that is in that area to become a Th1 cell. So it positive feedback. Another thing that the interferon gamma does is that, these cells are fairly selfish. They want to be the only star of the show. So whenever you have a Th1 response, your body essentially wants to turn off a Th2 response. So interferon gamma actually inhibits a Th2 response. So you amplify Th1, turn off Th2. Targets. What about targets, cellular targets here? Well, our interferon gamma is going to act on the classical macrophage. Remember those M1 macrophage? And it is going to hyperactivate them, make them those really crazed killers. Why? Because I'm trying to clear an intracellular infection that has been sort of troublesome. the macrophage in their normal states have trouble clearing the infection. So you hyperactivate them, making them, again, really good killers. And then finally, interferon gamma is going to allow B cells to undergo isotype switching to the IgG. So how do I remember that? Oh, well, think about it. Interferon gamma, got a G in it, is going to preferentially switch to IgG, which is going to be from the gamma heavy chain. Our next subset of helper T cells are going to be derived in response to parasitic infections. So the predominant subtype is going to be Th2. And recall that, hey, maybe this particular pathway will also be induced when I have things like type 1 hypersensitivities. So you could sort of put parasitic infections and type 1 hypersensitivities together as we go through this pathway. The predominant cytokine that is produced in response to a parasitic infection is going to be IL-4. Now, why is it that I go down a completely different pathway here? Well, the answer is pretty straightforward, is that with a parasitic infection, or I should say a majority of the parasitic infections, the pathogen's so large that it cannot be phagocytosed. So rather than engaging the cell-mediated immune response, you're more likely to go with a Th2 response where the actual destruction of the pathogen is going to be outside of a cell. So what does that mean to your body? Think about that. Well, that means that a Th2 response also has a higher potential to damage normal tissue in an attempt to clear the parasite. Because our parasites tend to be so large and not amenable to phagocytosis, you really don't see a high level of IL-12 production. And remember that IL-12 will drive cell-mediated immunity through Th1. Since I have no IL-12, the predominant cytokine being produced here is IL-4. And IL-4 is going to cause the upregulation of the transcription factor GATA3, which will drive differentiation of these cells down the Th2 pathway. So, what are some of the cytokines that are produced in response to parasitic infections? Well, a Th2 cell is way more complicated than a Th1, and it actually will secrete a variety of different cytokines, such as IL-4, IL-5, IL-10, 13, and finally TGF-beta. We're going to go through what each of these cytokines function as. While IL-4 alone can amplify a Th2 response, it is a combination of the activities of IL-4 and IL-10 that are able to inhibit a Th1 response. Again, going back to the idea that you want to preferentially have one response or the other. What about IL-5? And again, I'm hoping by this point y'all know what IL-5 does. IL-5 is going to drive development and activation of our eosinophils, which are going to be one of the principal cells of a Th2 response. Another cell that will be affected here are going to be our B cells. When I have IL-4 and IL-13, that is going to influence a B cell to undergo class switching to IgE. Now, IgE here is the predominant isotype, and it's really the one that I want you to focus on, but I do need you to know to a lesser extent, I might see some isotype switching to the sub-isotype form IgG4. But for your purposes, remembering IgE here is going to be a much more invaluable point. Another thing that I want to point out is that whenever I have a Th2 response, the macrophage that are going to be involved are going to be those of the M2 or alternative macrophage phenotype. And it should make a little bit of sense. Remember, what do those particular macrophages do? Well, they help in tissue repair. They clean up the mess. Whenever I have a parasitic infection again, it's large. It will directly cause damage to the tissue, and then when I'm trying to destroy it, I am going to have a lot of tissue damage from the destruction of the pathogen. So rather than have a macrophage come in that's going to do more killing, you bring a macrophage in that is essentially going to help clean up the mess. So that's another major difference between a Th1 response and a Th2. The newcomer to the T cell subsets is the Th17 grouping. Now, with the Th17 grouping, you are going to see an induction of these cells when you are having an extracellular, everybody listen, bacterial or fungal infection. This group of pathogens used to fall under Th2, so please make that mental note in your head that now we see a Th17 response. When I have infections with bacteria or fungi, the body is going to respond by, oh, shocker, producing all of those endogenous pyrogens like the IL-1 and the IL-6. The body also will secrete a couple of other cytokines like IL-23 and more TGF-beta. Do you guys remember where we saw TGF-beta before? Yeah, that was with our Th2. So Th2s will also produce some TGF-beta. But collectively, these cytokines are going to increase the expression of the transcription factor, I like calling it this, ROR gamma T, and that is going to induce differentiation of my T helper cells into Th17s. When a Th17 cell is activated, it in turn is going to secrete two different cytokines. The first one is really nice because it's named after the cell. I have a little IL-17. I also am going to produce some IL-22. Now, I'm notorious for taking IL-22 and exchanging it with IL-23. So I ask that you don't be me. Please remember that IL-23 is one of several cytokines that help induce differentiation into a Th17 cell, and the Th17 cells produce IL-22 in response to that activation. So what do these two cytokines do? Well, IL-17 does something that I think is pretty much common sense. It is going to increase the production of chemokines like IL-8. It's also going to increase other chemoattractants to bring more neutrophils into the site of infection. This makes perfect sense because you are dealing with an extracellular bacterial or fungal infection, and what cell is principally responsible for phagocytosing and eliminating these particular pathogens? Yeah, you're right. It's the neutrophil. So again, IL-17 bringing in neutrophils is really one of those no-duh moments. IL-22 is interesting in the fact that it is going to help stabilize interactions between endothelial cells. The purpose of this is to try to decrease the spread or movement of these pathogens through the tissue. And then both IL-17 and IL-22 are going to affect endothelial cells to start having immunologic function by secreting lots of antimicrobials into the environment. So Th17 cells, again, very straightforward mechanism. Always remember, though, that they are now the cells that are implicated in dealing with extracellular bacteria and fungal infections, whereas a few years ago, it was Th2. So let go of that memory and embrace the new. Okay, with this particular slide, we're not trying to confuse you, but yet to remind you that there are other subsets of T cells, especially our CD4 positive T cells. Now, our Tregs are principally produced during the development of our T cells within the thymus, but we want you to know that they are also formed in the periphery to a lesser extent upon activation within a secondary lymphoid tissue. Just as a reminder, our T regulatory cells directly inhibit Th1 cells, not Th2, not Th17, but Th1 cells. Please also recall what their surface markers are. Again, you should, anytime you see CD25, you need to be thinking about a regulatory T cell. And if you see any indication of the transcription factor, FOXP3, another big indicator. Now, Tregs like to secrete a lot of IL-10. Where did we just see IL-10 being produced? Do you remember? Dr. Moscatello remembers.
I remember.
You remember?
I do.
Okay, good. Well, for those of you who don't remember, Th2 cells also produce IL-10. Do you remember what the function is, Dr. Moscatello?
Oh, I'm thinking.
You're thinking? Oh, you know what I'm thinking it was? It was the inhibition of a Th1 response. So notice that, again, there's sort of a skewing here. If you are having a Th2 response or a Th17, you, in essence, are going to try to turn off a Th1. Also, you get production of TGF-beta. And there's another slide a little bit later on where we will talk about the impact of TGF-beta in the production of the isotype IgA. And even Tregs that are formed in the periphery, their functionality is pretty much the same. What they are trying to do is to turn off those self-reactive T helper cells, and again, specifically our Th1. And this portion of the lectures would not be complete unless we talk just a little bit about our cytotoxic T lymphocytes. Recall the information that Dr. Moscatello gave you about the cross-priming, where I can activate both CD4 positive T cells as well as CD8 positive T cells by one antigen presenting cell, like a dendritic cell. Also recall from the information I gave to you that both CD4 and CD8 require two independent signals. First is that primary signal through the T cell receptor, and then the co-stimulatory signal through the CD28. Now again, the major difference that we have is obviously the functionality. Our CD8 positive T cells have to be presented their peptide via MHC class 1, and you also need help from the T helper cells. Again, cytotoxic T lymphocytes are really not that good at producing their own IL-2, and that IL-2 is going to be extremely important again for T cell proliferation. So when you go through this, apply a lot of the same information to our CD8 positive T cells, but remember that they have their unique properties like the MHC class 1 and the lack of IL-2 production. Now that we've started to sift through some of these T cell subsets, probably be a good time to take a step back and ask ourselves, what kind of questions are we going to see on the step 1 with regards to these subsets? Classically, they like to talk about the different clinical presentations of an individual who has a Mycobacterium leprae infection. Now, in the U.S., we don't see a lot of leprosy unless somebody has come from a country where this is endemic. But on the step 1, you will see these clinical presentations, not so much from a microbiology standpoint, but more from an immunological standpoint. And really what they want you to understand is that the variations in the clinical presentations are based on the immune status of an individual, meaning do they have a predominantly Th1 response, which will generate cell-mediated immunity, or do they have more of a Th2 response? Rather than knowing all of the different presentations of leprosy, you really need to focus on the two ends of the spectrum, where we have tuberculoid versus lepromatous leprosy, and obviously with tuberculoid being the more benign of the two. So what is it that allows an individual to get infected with Mycobacterium leprae and only present with a tuberculoid? Well, one of the things you have to consider is their immune status. So a patient who has tuberculoid leprosy has a very solid Th1 response. And with a Th1 response, this is where you start immediately saying, okay, well, that's a cell-mediated immune response. I'm dealing with intracellular infections. Another thing you should recognize with a tuberculoid response, these individuals are going to form granulomas. They form these granulomas since it's more difficult to clear a mycobacterium infection. So these are some of the buzzwords that you might see in the clinical vignette, especially, and this is the one word that I always look for with these clinical vignettes, if they say the biopsy revealed a granuloma, I'm done. It's tuberculoid. It's a Th1 response. Okay, so why I pointed that out is that with all presentations of leprosy, you are going to see some extent of involvement with either the skin, you get those nodules, you will have effects of the peripheral nerves where you get the neuropathies, but this isn't going to be that helpful. So again, depending on where somebody is in their clinical presentation, there's going to be overlap. So you really are dependent upon the histological results. When you are dealing with somebody who has lepromatous is a very strong Th2 response. So I'm not getting that wonderful cell-mediated immune response, meaning I am not able to contain or eradicate the infection. And these individuals will have a much more severe presentation. What I really want you to focus on is in the clinical vignette. Again, they will talk about the biopsy of maybe one of the lesions, and rather than having the presence of a granuloma, they will describe diffuse acid-fast staining bacilli throughout the tissue. So those are the big two differences that you are going to see. Now, with lepromatous, you will get a low level of antibody production, but this is not going to be protective. So again, these individuals have a much more severe presentation than those with tuberculoid, and it is only individuals with lepromatous leprosy that are infectious to other humans.